cell signaling, and for further information, as always, you can see the individual article on it, and it's just on Wikipedia, so cell signaling. So cell communication or signaling is the ability of cells to receive, process, and transmit signals with its environment and with itself. Signals can be non-chemical, such as light, electric impulses, and heat, or chemical signals or ligands that interact with receptors, which can be found embedded in the cell membrane of another cell or located deep inside a cell. And just for a refresher, ligands, if we just go search right here, when I mentioned it earlier in the chemistry term. So a ligand is a substance that forms a complex with a biomolecule to serve a biological purpose. And the etymology stems from Latin to mean to bind. And in a complex or coordination complex consists of a central atom or ion, which is usually metallic, so something over here, and it's called the coordination center, and a surrounding array of bound molecules or ions that are in turn known as ligands or complexing agents. So that you have a bunch of ligands attached to this. And yeah, so these ligands, these attach but with a biomolecule that serves a biological purpose around this. Yeah, so fascinating stuff here. And here, just for reference, I'll just add the biomolecule definition just from Wikipedia. Okay, so make it like that. So basically, biomolecule or biological molecule is a loosely used term for molecules present in organisms that are essential to one or more, typically biological processes. Alright, so here's went back to the cell signaling one. Yeah, so basically signals can be non-chemical or chemical signals, or ligands that interact with receptors, or complexing agents, so ligands attached to other atoms making a complex, or attached to a central atom, yeah, or molecule or ion, etc. And these can be found embedded in the cell membrane of another cell, or located deep in the cell membrane of another cell, or located deep inside a cell. There are generally 4 types of chemical signals: autocrine, paracrine, juxtacrine, and hormones. Yeah, so let's go to the first one now. So autocrine, or autocrine, I'll just go with autocrine. So autocrine signaling, in autocrine signaling, the ligand, so molecules or ions that bind to a central atom forming a complex, affects the same cell that releases it. Yeah, so basically it's signaling to itself. So tumor, which are abnormal / excessive tissue growth cells, for example, can reproduce uncontrollably because they release signals that initiate their own cell division. Yeah, so this is signaling to itself. And then paracrine. In paracrine signaling, the ligand diffuses to nearby cells and affect them. For example, brain cells called neurons release ligands called neurotransmitters that diffuse across a synaptic cleft, which is a small space between neurons, to bind with a receptor on an adjacent cell, such as another neuron or muscle cell. So yes, in paracrine signaling, you have the ligand diffuse, or the complexing agents get released from that atom and then go over to another cell that's nearby, and then that affects how that other cell responds, and hence you're signaling information to it. So MES note: a synapse is a structure that permits a neuron or nerve cell to pass an electric or chemical signal to another neuron or to the target effector cell. Going for this, so chemical synapses. Yeah, there's different types of synapses. So you could either do this one, pass an electric signal or chemical signal, or other types of signal. So a chemical synapse, chemical synapses are biological junctions through which neuron signals can be sent to each other and to non-neural cells, such as those in muscles or glands. And chemical synapses are not the only type of biological synapse. There's electrical and immunological synapses. Those also exist. Without a qualifier, however, synapses commonly refers to chemical synapses, or to a chemical synapse. So it's fascinating stuff. Let's continue further, see an illustration of this right here. So right here, the connection, make it a bit smaller. Yeah, so here, so the connection linking neuron to neuron is the synapse. Signal flows in one direction from the presynaptic neuron to the postsynaptic neuron via the synapse, which acts as a variable attenuator. They basically adjust the strength of the signal getting through. So in brief, the direction of the signal flow determines a prefix for the involved synapses. So post is after, pre is before the signal is gone through. So here's to distinguish between pre and post synapse. And again, the idea of these synapses is a structure that permits a neuron or nerve cell to pass through. So here would be the presynaptic neuron. So neurotransmitter transmission, which is again is a ligand. And neurotransmitter is a ligand, so brain cells called neurons release ligands called neurotransmitters as signals that diffuse across a synaptic cleft. So there's a synaptic cleft between these two neurons, and this would be considered the pre-synaptic neuron and neurotransmitter transporter. Yeah, so this is the neurotransmitter transporters from here, and then the receptors at the bottom, and there's a synaptic cleft in between. So it goes from the presynaptic neuron and then to the postsynaptic neuron. So the pathway of these ligands or signals is coming across there. So neurotransmitters is the transporter is over here. Then there's the receptor. Just the receptor, or this side right here. Yes, basically the ligands go transport over to here, and it's nearby. So paracrine. So going back here. So paracrine signaling the ligand diffuses to nearby cells. And then autocrine is inside, or it's to the same cell. All right, let's continue further. And now this is an attenuator, because basically, going back here, so signal flows in one direction from the presynaptic neuron to the postsynaptic neuron via the synapse, which acts as a variable attenuator. And essentially, so the synapse acts like that. And this variable attenuator is an electronic device that reduces the power or rate of energy transfer of a signal without appreciably distorting its waveform. So if you have, let's say, a waveform like this coming through, but the rate is very, very fast, what you do is just basically slow it down. So if this is going, because this is with time, so it's the rate. So if you have something super fast like this, what you'll do is slow it down so that you'll have something like this slowly going through, and so you're reducing that power, the rate of energy transfer. But but the waveforms are without appreciably distorting its waveform. So these are similar. These are similar waveforms to these. Yeah. So basically, it would let this through. So it acts as a variable attenuator. So this is energy is going through, and then it would just slow down how fast it's going in. All right. So the next one is juxtacrine signaling, and remember there was 4 types here. Scroll up here. So there are generally, just put that word, generally 4 types, probably some special cases elsewhere. So generally 4 types of chemical signals: autocrine, paracrine, juxtacrine, and hormone. So now let's look at juxtacrine. So in juxtacrine signaling, there is direct contact between the signaling and responding cells. And yeah, so that's pretty much all you need to know in this case. But yeah, so the difference is paracrine is nearby cells, so such as neurons. So there's a cleft right here. But then with this is in direct contact with it, and you could view it as a muscle attached to another cell, or just basically you have just two cells connected directly through there's a membrane there, and then you have molecules sent across there. So basically, it's direct contact with the other ones. So yeah, you have another one. So and then the difference with the paracrine is that it's nearby. So there's a difference you're sending through, and the other ones are in direct contact with them and transmitting signals. And yeah, going further. So finally, hormones are ligands that travel through the circulatory systems of animals or vascular systems of plants to reach their target cells. So yeah, so these travel all the way around to far distances. So I must note the circulatory system, also called the cardiovascular system or the vascular. These are channels, vascular channels for conveying fluid. System is an organ system that permits blood to circulate and transport nutrients such as amino acids and electrolytes, oxygen, carbon dioxide, hormones, and blood cells to and from the cells in the body to provide nourishment and help in fighting diseases, stabilize temperature and pH, and maintain homeostasis. And yeah, so homeostasis is the state of steady internal physiological and chemical conditions maintained by living organisms. And we'll go over some of these other terms. So an electrolyte is a substance that produces an electrically conducting solution when dissolved in a polar solvent such as water. Yeah, so this is in the transport the circulating transport nutrients such as amino acids and electrolytes. So you have a substance that produces an electrically conducting solution. The dissolved electrolyte separates into cations and anions, which disperse uniformly through the solvent. Electrically, such a solution is neutral. If an electric potential is applied to such a solution, the cations of the solution are drawn to the electrode. So cations is plus charge. And yeah, so basically you have cations plus and minus, and they're all separate. They're dispersed uniformly. So again, similar to a salt. We view it as salt in solution. And then when you apply an electric potential or electric pressure, basically a bunch of charges of one kind on one side and the other one on the other, the cations of the solution are drawn to the electrode, which is a conductor in contact with a non-metallic part of circuit. Yeah, so essentially a conductor where it allows electrons to flow through or current to flow through, but this is a non-metallic part of the circuit. And yeah, so then the cations go to the electrode that has an abundance of electrons. Yes, so basically electrons on one side, so go into the minus, while the anions, which are minus, are drawn to the electrode that has a deficit of electrons. So basically it has a relative charge, relative plus charge, like that. All right, let's just erase these now. And going further, so the cardiovascular systems, remember this is the circulatory, also called the cardiovascular system. The cardiovascular system, from Latin words meaning heart and vessel, system comprises the blood, heart, and blood vessels. It includes the pulmonary circulation. This is involving the lungs, yeah, which is a, quote, loop through the lungs where blood is oxygenated, and systemic circulation, which is a, quote, loop through the rest of the body to provide oxygenated blood. So basically this one here, blood goes to the lungs to get oxygen, and then then there's a second loop that goes to the rest of the body to provide the body with oxygenated blood, hence why oxygen is needed, yeah, to breathe and live, and etc. And oxygen is usually a byproduct of photosynthesis. Yeah, hence the waste of plants is what humans need to, or animals need to live. So the cardiovascular system of humans are closed, meaning that blood never leaves the network of blood vessels. All right, let's continue further. So here is an illustration. This is circulatory system, but it's ignoring the lymph system. So this is a cardiovascular system. Yeah, so here's just a whole bunch of stuff here. So there's the blue. This one is bringing deoxygenated blood, and the red is oxygenated blood. And across. So basically the human circulatory system, simplified, red indicates oxygenated blood carried in arteries, so this is from the heart, and then blue indicates deoxygenated blood carried in veins, so these are blood vessels, and arteries are from the heart, so with oxygen. Blue is towards the heart, in veins, so to the heart, and these are deoxygenated, so it's lost its oxygen because it's given to the body. And capillaries, which join the arteries and veins, and the lymphatic vessels are not shown. Here's a whole bunch of so you have the red pumps into it here. So this one is a pulmonary one. See these spikes all the way out, like just outside here. So let's draw it here. These in here, that's pulmonary system going to the lungs, getting oxygen from them. So the blue goes to the deoxygenated goes to the lungs, and then it brings back oxygenated blood to the red part right here, and this whole arteries go across to the whole body. And then the capillaries are what joins arteries and veins, so it goes all the way to the body. The capillaries, but they're not shown here, and they would bring in from inside the blue and all the way back to the heart, which gets to the lungs and back again, and just keeps pumping back and forth. And now let's briefly discuss the lymphatic system. So the lymphatic system, or lymphoid system, is an organ system in vertebrates that is part of the circulatory system and the immune system. It is made up of a large network of lymphatic vessels, lymph nodes, lymphatic or lymphoid organs, and a lymphoid tissue. But yeah, so just note that. So it's part of the circulatory system, but the cardiovascular one is specifically about the heart and just basically system comprises of blood, heart, and blood vessels. But circulatory system is also called cardiovascular system, and this one is about blood transporting and nutrients, etc. But so the lymph system is still a part of the circulatory system. So to be more technical, this should include the lymphatic system, and then to be more specific, the cardiovascular would be just the heart, just a blood setup where it's blood, heart, and blood vessels. and then the lymph would be a second part of the circulatory system. And that is for so this basically is an organ system. It's also part of the immune system, and is an organ system, and part of the circulatory system and immune system, and we'll get to that right now. So it is made up of a large network of lymphatic vessels, lymph nodes, lymphatic or lymphoid organs, and lymphoid tissues. The vessels carry a clear fluid called lymph, and the Latin word lympha refers to the deity of fresh water, or called lympha. Yeah, and this carries it towards the heart. So the vessels carry this fluid called lymph to the heart. So unlike the cardiovascular system, which again the circulatory system is also called the cardiovascular system, just in often usage, but to be more specific, so cardio is not the circulatory, it's part of the circulatory, and lymphatic is part of the circulatory. And so basically these are all parts of this one. It's going further. So unlike the cardiovascular system, the lymphatic system is not a closed system. The human circulatory system processes an average of 20 liters of blood per day through capillary filtration, which removes plasma from the blood. Roughly 17 liters of the filtered blood is reabsorbed directly in the blood vessels, while the remaining 3 liters are left in the interstitial fluid, which is the fluid between blood vessels and cells. And so one of the main functions of the lymphatic system is to provide an accessory return route to the blood for the surplus of 3 liters. So technically, it's still all part of the circulatory system. But the cardiovascular one is specifically on that closed loop of the heart. So that was the heart, lungs, and blood. So heart. Let's go. This one is for heart. It's blood, heart, and blood vessels. And then excluding the lymphs, which is all around. And those provide an extra 3, provide an accessory route or an extra route, return route to the blood. Of the extra 3 liters. So basically, right here, so the circulatory system processes 20 liters of blood in humans per day through capillary filtration, which removes plasma from the blood. And then roughly 17 liters of the filtered blood is reabsorbed directly into blood vessels, while the other 3% are still all around. It is in the interstitial fluid, which is between the blood vessels and cells. And the function of the lymphatic system is to provide, or one of the main functions is to provide an accessory route, return of the blood, accessory return route to the blood for the surplus 3 liters. So this is one of the main functions. So still all pretty much part of the circulatory / blood flow system. So it's just part of the blood wants to get back of it. And here is the lymphatic, human lymphatic system. And here's a bunch of, so there's a spleen as part of it, and there's a bunch of nodes right here, and they all function in cell signaling and other types of biological functions. And then this provides return route back to the blood. Back to the blood and / back to the heart, because all the blood vessels eventually go back to heart. So it's all moving that clear fluid called lymph towards the heart. And going further, so lymph, from Latin lympha, meaning water, is the fluid that flows through the lymphatic system, a system composed of lymph vessels or channels. So a bunch of lymph vessels all across here. And so it's a system composed of lymph vessels or channels and intervening lymph nodes, which are small kidney-shaped organs whose function, like the venous system, is to return blood from the tissues to the central circulation. So you have a bunch of nodes here, and it goes over here, then it also redirects all the way back to the heart and blood vessels, hence all the way back to the heart. So at the origin of the fluid return process, interstitial fluid, which is the fluid between cells and all body tissues, enters lymph capillaries. This lymphatic fluid is then transported via progressively larger lymphatic vessels through lymph nodes, where substances are removed by tissue lymphocytes, which is a type of white blood cell, which are part of the immune system. We'll get to that later in this video. And circulatory lymphocytes are added to the fluid before emptying ultimately into the right or left subclavian vein, where it mixes with central venous blood or deoxygenated blood. So basically goes into the deoxygenated into the vein, which is where the blood goes towards deoxygenated blood goes back to the heart. Because it is derived from interstitial fluid, from which blood and surrounding cells continually exchange substances, lymph undergoes continual change in composition. It is generally similar to blood plasma, which is the fluid component of blood. And then lymph returns proteins and excess interstitial fluid to the bloodstream. Lymph also transports fats from the digestive system to the blood, just fasting and stuff. So it pretty much has a lot of very important roles that assist with the closed loop system of the blood flow, and then you have bringing in from the pretty much all the excess water and other stuff going back to the blood. So the subclavian vein, and this is over here where substances are removed by tissue lymphocytes, and circulating lymphocytes are added to the fluid before emptying ultimately into the right or left subclavian vein. So this one here is a paired large vein, one on either side of the body, that is responsible for draining blood from the upper extremities, allowing this blood to return to the heart. And we can actually see the subclavian vein in this diagram right here. So going to the circulatory system one. So here's subclavian vein A and V. So artery is over here, is the red, and then vein is V. So right there is the V. So this one right here, this big giant one, that's subclavian vein, but that's the right one, because is your right hand, and there's the left. So you got one here, left and right, subclavian vein. So there's those really giant veins there. And so basically goes, the lymph is all directed over here. A lot of it is directed here, and then pushed over back to the vein systems, and then going to the heart. And so you have other ones from the bottom of the body, and this other ones from the top. So the subclavian vein is responsible for draining blood from the upper extremities, allowing this blood to return to the heart. So such as your arms, shoulders, and so on. Lymph capillaries or lymphatic capillaries are tiny, thin-walled microvessels located in the space between cells, except in the central nervous system and non-vascular tissues, which serve to drain and process extracellular fluid, or more specifically interstitial fluid. We'll get over types of fluids soon. So now let's look at, just before we get to the lymph capillaries, let's look at basic capillaries right here. So here's a simplified illustration of a capillary network. And remember, these are just tiny blood vessels that connect the arteries to the veins, and also serves as a outlet and inlet to get fluid from or to the surrounding body tissues. And then there it connects the artery. This is the oxygenated blood from the heart, and then this is the vein deoxygenated blood to the this is back to the heart. So this is from the body into the heart. There's a capillary. There's the small network of micro blood vessels connecting these two. And here's lymph capillaries in the tissue spaces, and you can see them here. These are in between even the capillaries right there. And so here's a diagram showing the formation of lymph from interstitial fluid, labeled here as tissue fluid. So there's tissue fluid right here. And note how tissue fluid is entering the blind ends of interstitial fluid. These blind ends are just these right here. These I assume that's what they're coming. So this is coming in from these quote blind ends. I don't know why they use that terminology. Basically going over there, which has no direction, and then all of a sudden you have these all these network of capillaries over there. So they could have just said from the free end over here, and these are lymph capillary in green. Notice how there's even in between the regular capillaries right there for the blood vessels. And over here, this is an arteriole, or actually pronounced arteriole. Yes, arteriole, and this is a venule, or a venule. And these ones right here, I'll get to those in a bit. But here's a lymphatic vessel right here, and then here's a bunch of tissue cells over there. So interstitial fluid is the ones in between the cells. And this arteriole has oxygenated blood coming in from the heart, and this is the venule. This is towards the heart, so deoxygenated blood. And just a note here, an arteriole is a small diameter blood vessel in the microcirculation that extends and branches out from an artery and leads to capillaries. Arterioles have muscular walls, usually only 1 to 2 layers of smooth muscle cells, and are the primary site of vascular resistance. The greatest change in blood pressure and velocity of blood flow occurs at the transition of arterioles to capillaries. So yes, there's a junction here, and I'll show you an image soon. A venule is a very small blood vessel in the microcirculation that allows blood to return from the capillary beds to drain into the larger blood vessels, the veins. So here's a diagram to illustrate that. So there's an arteriole, and there's an artery. So this is has high resistance here. It's going here. This is a high resistance area that can slow down the blood flow from the arteries right there. And then there's the tissue cells. There's the venule. It's coming back in. It goes in here, goes through there, goes all the way across, and goes in here. And then there's the vein. So the vein is the big one. The venule is the connection, and then the capillaries are the small network of blood vessels. The illustration of blood vessels including artery, arteriole, capillaries, vein, and venule. And now let's discuss the interstitial fluid. So interstitial fluid is the body fluid between blood vessels and cells, containing nutrients from capillaries, which are the small blood vessels connecting arteries and veins. Those are just right here. By diffusion and holding waste products discharged out by cells due to metabolism. And you can see that you have a whole bunch of tissue cells there. And the interstitial fluid is between blood vessels and those cells. It gets released, including waste products, and those can be stored in that fluid. And here's a distribution of the total body water in mammals between the intercellular compartment, which is within cells. This is the intracellular compartment within cells, and the extracellular compartment, which is outside of cells, which is in turn subdivided into interstitial fluid and smaller components such as the blood plasma and the cerebrospinal fluid, which surrounds the brain and spinal cord, and lymph, which makes up a small percent of the interstitial fluid. Yeah, so here's a diagram. So the intracellular fluid is inside cells, is 67% of the body water in mammals, approximately. Then extracellular fluid outside of cells, 26% of that is going to be interstitial fluid, as between blood vessels and cells. And then the intravascular fluid, which is inside the blood plasma or in the blood vessels, it's 7%. And then the spinal cord and brain, that's a cerebrospinal fluid, that's less than 1%. Fascinating stuff. And then lymph makes up a small percent of the interstitial fluid, so a percent of this one. More note on this body water. So in lean, healthy adult men, the total body weight is about 60%, or 60 to 67% of the total body weight in men. It is usually slightly lower in women, 52 to 55% body water. The exact percentage of fluid relative to body weight is inversely proportional to the percentage of body fat. So the more body water, the less fat. The more fat, the less body water. Fascinating stuff. All right, so now let's go over a lymph node. So a lymph node or lymph gland is a kidney-shaped organ of the lymphatic system and the adaptive immune system. A large number of lymph nodes are linked through the body by the lymphatic vessels. They are major sites of lymphocytes that include B and T cells. We'll get over those later in this video. This is part of the immune system. Lymph nodes are important for the proper functioning of the immune system, acting as filters for foreign particles, including cancer cells, but have no detoxification function. So here's a whole bunch of parts of this lymph node. So a diagram showing major parts of a lymph node. Yeah, so it looks like a kidney or kidney bean, and yeah, this is just for diagram purposes. So a bunch of vessels connected here. This is blood vessel entering here, the hilum, or actually I think that's pronounced hilum. And then lymphocytes and outflowing lymph and so on. So it goes in and out. And I just want to illustrate just the zoom in of that. And these are just a bit beyond, this video. That's just a bit more detailed, and we need to go into it right now. But we will go over lymphocytes and B and T cells later in this video when we discuss the immune system. So once a ligand binds with a receptor, and again, note that we're back into the signaling discussion. So once a ligand binds with a receptor, it can influence the behavior of another cell depending on the type of receptor. And let's just get a refresher right here on the type of type of signaling. So we have here, we had therefore generally four types of chemical cell signals: autocrine, paracrine, juxtacrine, and hormones. And then autocrine is the ligand, which again is molecules or ions that bind to a central atom, forming a complex. Yeah, so the autocrine affects the same cell that releases it, so just it signals to itself. And then paracrine is to nearby, so diffuses to nearby cells and affects them. And going over like synapses and so on. So there's a separation point right here, a junction, or synaptic cleft right here. And then in juxtacrine signaling, there's direct contact, so that the cells are actually touching each other. and then they're signaling and responding accordingly. And finally, hormones are ligands that travel through the circulatory system of animals or vascular systems of plants to reach their target cells. And then I went over a tangent on circulatory system, lymph nodes, and so on. Let's go back over to here. Let's get our brain back into it. And also because I'm filming this several days apart, I guess I would pause it whenever I get to X amount of time and then go do some other work and then come back to it. And hence why it's going to be very, very long. So once a ligand binds with a receptor, it can influence the behavior of another cell depending on the type of receptor. Yeah, so for example, or for instance, neurotransmitters that bind with an inotropic receptor, which is an ion channel protein, can alter the excitability or ease of response of a target cell. Other types of receptors include protein kinase receptors, e.g. receptor for the hormone insulin, and G protein coupled receptors, and G protein coupled receptors. And these are just the type of proteins, so G protein are coupled to them. And activation of G protein coupled receptors can initiate second messenger cascades. And we'll go over that in a bit in the MAS note. In the process by which a chemical or physical signal is transmitted through a cell as a series of molecular events is called signal transduction. Yeah, so you're going from chemical or physical signals transmitted through a cell as a series of molecular events or reactions or interactions. It's called signal transduction. So it's basically changing form and being transmitted. Alright, so now let's look at the MES note and basically dissect all these random terms. So a kinase is an enzyme that catalyzes the transfer of phosphate groups from high energy phosphate donating molecules to specific substrates. And that's as other types of receptors include protein kinase right there, such as a receptor for the hormone insulin. A protein kinase is a kinase which selectively modifies other proteins by covalently adding phosphates to them, aka phosphorylation, as opposed to kinases which modify lipids, carbohydrates, or other molecules. So a kinase is just an enzyme that catalyzes the transfer of phosphate groups from high-energy phosphate-donating molecules to specific substrates, and a protein kinase does this to a protein as opposed to lipids, carbs, or other molecules. And here's the illustration, a general scheme of kinase functions. So, for example, here's a protein, and we'll have a protein kinase, and then there's a phosphate group right here. So here's phosphate groups. This is ATP. This is adenosine triphosphate, and there's adenosine diphosphate. There's two phosphates. This is three phosphates, two phosphates. So either or actually in this case, it will take the ADP right here, and it will take one phosphate. So we'll take this one right here and move it over there, and then now this is missing. So it's missing one here. So it's fascinating stuff. So the ATP turns into ADP, and then it takes that phosphate and then phosphorylates the protein, so it connects right to it. So very interesting stuff. So insulin, because in this setup here, receptor for the hormone insulin. So insulin, from Latin insula or island, is a peptide hormone produced by beta cells of the pancreatic islets or inlets. Actually, it's islets. It is considered to be the main anabolic hormone of the body. It regulates the metabolism of carbohydrates, fats, and protein. And this does this by promoting the absorption of glucose from the blood in liver, fat, and skeletal muscles. And a peptide hormone right here, or protein hormone, are hormones whose molecules are peptides or proteins, respectively. The latter have longer amino acids, the proteins, longer amino acid chains lengths than the former. So peptides is pretty much just short amino acid chains. Proteins are long amino acid chains. The pancreatic islets, or islets of Langerhans, I think that's how you pronounce it, are the regions of the pancreas that contain its endocrine, which is hormone-producing cells. And the pancreas is an organ of the digestive system and endocrine system of vertebrates. In humans, it is located in the abdomen behind the stomach and functions as a gland. The pancreas is a mixed or heterocrine gland. It has both an endocrine and a digestive exocrine function. 99% of the pancreas is exocrine and 1% is endocrine. As an endocrine gland, it functions mostly to regulate blood sugar levels, secreting the hormones insulin, And yeah, I think this is pronounced glucagon, and then this is somatostatin, or somatostatin, and pancreatic polypeptide. As a part of the digestive system, it functions as an exocrine gland, secreting pancreatic juice into the duodenum through the pancreatic duct. This juice contains bicarbonate HCO3-, which neutralizes acid entering the duodenum from the stomach, and digestive enzymes, which break down carbohydrates, proteins, and fats in food entering the duodenum from the stomach. And now let's just get a refresher, actually, mainly for me. Let's look at a gland from my earlier part here. So a gland is a group of cells in an animal's body that synthesizes substances such as hormones for release in the bloodstream, that's endocrine gland, or into cavities inside the body or its outer surface, basically releasing it elsewhere. If you're going back to the bloodstream, that's endocrine. Releasing outwards, that's exocrine. So that's fascinating stuff. So let's go back over to it. That's going to be right here is pancreas, like that. Yeah, so basically as an endocrine gland, so this pancreas functions, 99% of it is as a exocrine gland, so releasing substances outside of itself to other cavities in the body or to outside of the body, and 1% is endocrine inside the bloodstream. Yes, so let's continue further right here. So a pancreatic juice, and that's this part right here. So as part of the digestive system, it functions as an exocrine gland, secreting pancreatic juice into the duodenum through the pancreatic duct. Yeah, so pancreatic juice is a liquid secreted by the pancreas, which contains a number of digestive enzymes, including these enzymes, tryptosinogen, or trypsinogen, and now this one here. This is a chymotrypsinogen. This one elastase, and this is carboxypeptidase. Actually, I think it's pronounced carboxypeptidase. Yeah, carboxypeptidase, and pancreatic lipase, and this is nucleases, and it's amylase. That's an exercise, and it's in of itself pronouncing this and Google searching the pronunciation. The pancreatic duct, or duct of Wersan. Yes, interestingly named after this. So duct of Wersan. Wersan, also the major pancreatic duct due to the existence of an accessory pancreatic duct, is a duct joining the pancreas to the common bile duct. This supplies it with pancreatic juice from the exocrine pancreas, which aids in digestion. The common bile duct, sometimes abbreviated as CBD, is a duct in the gastrointestinal tract of organisms that have a gallbladder. In vertebrates, the gallbladder, also known as a cholecyst, is a small hollow organ where bile is stored and concentrated before it is released into the small intestine. And bile, from Latin bilus or bilus, yeah, I think it's pronounced bilus, or gall, is a dark green to yellowish brown fluid produced by the liver of most vertebrates that aids the digestion of lipids in the small intestine. In humans, bile is stored continuously by the liver, and it's called liver bile, and stored and concentrated in the gallbladder. After eating, this stored bile is discharged into the duodenum. Yeah, or duodenum. Yeah, so fascinating stuff. So there's a diagram of it. So pancreatic islets are groups of cells found in the pancreas that release hormones. And here is pancreatic tissue right here. So this is a zooming in of the pancreas right here. And if you zoom in, you have right here, these right here, pancreatic, acini, or pancreatic acini. These have the exocrine cells. It's released outside, and then these are pancreatic islets with endocrine cells inside here. And these are inside this little zooming in right here. So this part right here. Yes, fascinating stuff. There's pancreas, and there's the location in the human body there. And there's the gallbladder right there. There's the common bile duct, and there's an accessory pancreatic duct right there, or secondary one. And there's a duodenum is opening right here, a papilla. And there's a duodenum right here connected to the intestines from the stomach. And there's a pancreatic duct there, and there's a pancreas. Yeah, so the gallbladder right here is a small whole organ where the bile is stored and concentrated before it is released, and the bile is released into the small intestine, and the bile or gall is a dark green fluid, yellowish to brown fluid produced by the liver of most that aids in digestion of lipids in the small intestine. Yeah, so this stores bile or gall, and then it releases it into the small intestine to aid in digestion. Let's continue further right here. So here's pancreatic islets from a mouse in a typical position, close to a blood vessel, insulin in red, nuclei in blue. Yeah, so there's the insulin right there in red, and the nuclei in blue there. And it's fascinating stuff. And then insulin, again, is, let's just refresh on that. So insulin is a peptide hormone produced by beta cells of the pancreatic islets. It is considered to be the main anabolic hormone of the body, or this is building up. Yes, a hormone also signals to build up or regulates the metabolism of carbohydrates, fats, and proteins by promoting the absorption of glucose from the blood in liver, fat, and skeletal cells. So it is the main anabolic hormone, or the main building up hormone, to signal to just produce more molecules. And a side note here, this is visualized using immunofluorescent microscopy. Colors are red is insulin antibody, blue is DAPY equals nuclei. Nuclei, green is unspecific anti-mouse secondary antibody. Stains mostly intercellular matrix. So basically a bunch of stains, and using antibodies. We'll get over those later in this video. Actually, we already had covered antibodies, actually. Yeah, let's just go back to the antibody, but first is DAPY. We had this, we already went over this earlier. We already came across, let's just do a search, and DAPY, this was from the earlier image right here. So DAPY, the nucleus is stained, so this blue nucleus is stained with DAPY fluorescent stain that binds strongly to adenine, thymine rich regions in DNA, and that's where it's inside the nucleus. So then it binds to it, and you can stain it, and you could get this bluish color there. And yeah, I'm assuming that the actual stain is blue, but nonetheless it could be edited. But just to show the contrast there, it's fascinating stuff. And that is DAPY. Let's go back to the antibody. Just do a brief antibody. Yeah, so antibody AB, also known as immunoglobulin Ig, is a large Y-shaped protein used by the immune system to identify and neutralize foreign objects such as pathogenic bacteria and viruses, and so on. So basically, there's the antibody right here, and then it can recognize different antigens that react with it, such as bacteria and so on. Yeah, so each antibody binds to a specific antigen, and interaction similar to a lock and key. Alright, so let's get back to here. So basically, you have the blue dye here for the DNA regions there, and then there's the insulin right here in red. So fascinating stuff. And dimensions real width 326 micrometers or microns. This is a millionth of an inch. Of a meter. And this is generated in the Solomina lab. This is at the Paul Langerhans Institute in Dresden. And going further, so immunofluorescence right here, microscopy, is a technique used for light microscopy with a fluorescence microscope, and it is used primarily on microbiological samples. This technique uses the specificity of antibodies to the antigen to target fluorescent dyes to specific biomolecule targets within a cell, and therefore allows visualization of the target molecule, visualization of the distribution of the target molecule through the sample. And recall that fluorescence, let's just do a quick check. So fluorescence is the emission of light by a substance that has absorbed light or other electromagnetic radiation. Just fascinating stuff. So let's just go back over here. Yeah, so back to over here. So this uses an immunofluorescence technique that uses antibodies that are specific to their antigens to target fluorescent dyes. It's basically manipulating the antibodies, their specificity, and to target dyes, and then you can basically shine light where it absorbs and then emits other light that you can detect. And then you could use that to visualize the distribution of a target molecule throughout the sample, such as this right here. There's a nuclei, and there's the insulin. Right, going further, so G protein coupled receptors, that is over here. We discussed that here. So activation of so this one here. Other types of receptors include protein kinase, so receptor for the hormone insulin, and G protein coupled receptors, and that uses protein kinase. So let's just go over that quickly. So G protein coupled receptors, GPCRs, are cell surface receptors that detect molecules outside of the cell and activate cellular responses. And G proteins, also known as guanine nucleotide binding proteins. This is a guanine nucleotide binding proteins, are family of proteins that act as molecular switches inside cells and are involved in transmitting signals from a variety of stimuli outside of a cell to its interior. It's just proteins with specific functions. In this case, signals transmitting from outside of a cell to its interior. And secondary messengers, going back over here again. So there's a lot of back and forth. So right here, activation of G protein coupled receptors can initiate second messenger cascade. So you get signals from outside of the cell, from the G proteins. It can transmit that into the cell, and then you have a secondary messenger cascade, or in this case, second messengers are intracellular signaling molecules, so inside, released by the cell in response to exposure to extracellular signaling molecules. So you have the first messengers. The G proteins, the first messengers over there transmitting from outside to inside. Now you have a secondary message being transmitted inside the cell. So here is a complex diagram here. So signal transduction pathways that lead to a cellular response. And remember, signal transduction is just a series of molecular events right here. So it's a process by which a chemical or physical signal is transmitted through a cell as a series of molecular events. It's called signal transduction. So a whole series of molecular events. And here is a complicated diagram right here, just an in-depth diagram right here. So you have cytokines and other molecules going in. There's a receptor. There's a survival factors right here. Receptor. These are hormones, transmitters, and so on. GPCR. This is G proteins right there. Cell surface proteins. You have other kinds right here. Growth factors, extracellular matrix connecting to these. Basically a whole bunch of stuff inside there. And then here is a lot of different pathways going on. So either this signal right hormone comes in here, G protein, and it can go different directions. And there's right here, this is inside, I believe, the cell nucleus, or I think this might be just more general, related to possibly to the cell nucleus. But anyways, this goes into gene regulation, cell proliferation. And you have a whole bunch of other pathways, all different chemical reactions and molecular events, and different changing of molecular characteristics and specific atoms, elements of it, all being interacted all in this cell right here. And then all leading to different cellular responses. You can either cell proliferation, either a duplicated self, or you can even go to apoptosis. So you can go over to there, which is a programmed cell death, and then compare that with necrosis, which is traumatic cell death. So this could be eventually at its end of its lifetime. It can signal pathways will say, okay, we have had enough, and you just program cell death. It just kills itself, or just blows up, and or just dies. So that's fascinating stuff. And necrosis is traumatic cell death, so something affecting it and forcing it to die.